Dear students, welcome to EPG Pathshala. I am Dr. Y.S. Rajput, former Head of Division, Animal Biochemistry Division, National Dairy Research Institute, Karnal. Today I am going to teach you technique of polyacrylamide gel electrophoresis under the paper Biochemical Techniques. Fundamental aspects of the technique have already been covered in module under electrophoresis basic concepts. In that module, we have covered concepts of pH, concept of pKa, Henderson-Hasselbalch equation, electrophoretic velocity, electrophoretic mobility, amount of heat generated in electrophoresis, and its influence on performance of electrophoresis. Today, I will be explaining one of the specialized electrophoretic technique, that is acrylamide gel electrophoresis. There are two types of acrylamide gel electrophoresis. First one is referred as polyacrylamide gel electrophoresis, and in short it is called PAGE, P-A-G-E, while the other one is referred as sodium dodecyl sulfate polyacrylamide gel electrophoresis, or in short SDS-PAGE. The technique is highly used for the separation of the protein molecules. The separation is based on the size of the molecule. There are two different designs in which the electrophoresis can be carried out depending upon the two different units. If the unit is horizontal one, the electrophoresis is referred as the horizontal gel electrophoresis. If the unit is vertical one, then electrophoresis is referred as the vertical gel electrophoresis. So it should be clear that two different designs are available for conducting the electrophoresis, and accordingly these are being referred. If we see the concepts used in the SDS-PAGE electrophoresis, this is explained in terms of its capability to resolve the proteins in the gel. And when we say vertical gel electrophoresis, the gel used is the acrylamide because it is more elastic, and so elasticity helps in removal of the gel very conveniently. SDS-PAGE can be carried out for the determination of the purity of the protein molecule. Also, it can be used for determination of the molecular weight of the proteins. So this technique is also a prerequisite to the Western blot, which is used for the characterization of the protein molecule. Thus, this technique is routinely used in protein laboratory and all subjects of the life science. In this presentation, our focus will be on to understand supports available for separation of proteins in electrophoresis. Also, we will make clear that the difference between polyacrylamide gel electrophoresis, that is PAGE, and also the sodium dodecyl sulfate polyacrylamide gel electrophoresis, that is SDS-PAGE. How this polyacrylamide gel is prepared, this will be also discussed in the presentation. We have to see that what could be the potential applications of the SDS-PAGE and what precautions one should take it for performance of the SDS-PAGE, so that students do not face the problem while they use this particular technique on their bench. Arne Tesselius in year 1937 developed Tesselius apparatus for electrophoresis, where charged particles were separated in free solution and electrophoresis is referred as moving boundary electrophoresis. This was associated with the problem of diffusion of separated molecules and convection current. To overcome these problems, separations were subsequently attempted on supports such as paper and gel. This resulted in separation of particles in the form of zone, and the technique is referred as zone electrophoresis. Filter paper, cellulose strips, and gel provide mechanical support, and also these are porous. Porous nature of these supports allows movement of buffer ions and sample during electrophoresis. Support also reduces convection current. Separation of these supports also reduces diffusion of particles. These properties of support result in separation of charged particles as sharp zone, and electrophoresis is referred as zone electrophoresis. Because of porosity limitation, relatively small molecules such as amino acids, nucleotides, peptides are separated on paper. Acrylamide gel, star gel, and agar gels are used for separation of protein. Oliver Smith in 1955 used star gel electrophoresis for separation of protein from human tissue. Agar gel can be used for separation of large size proteins. Acrylamide gel is preferred over star gel since acrylamide gel of desired pore size can be easily prepared in the laboratory. In both the techniques, gel is prepared by polymerization of acrylamide. Mixture of proteins can be separated in both polyacrylamide gel electrophoresis and sodium dodecyl sulfate polyacrylamide gel electrophoresis. In PAGE, electrophoretic mobility is dependent on net charge on each of the protein, which of course is dependent on composition of protein and pH of buffer used in electrophoresis. Since all proteins in mixture are separated simultaneously, potential gradient and to large extent frictional coefficient will be constant. However, in case of sodium dodecyl sulfate polyacrylamide gel electrophoresis, electrophoretic mobility is dependent on molecular size or mass of protein. In this technique, charge to mass ratio for all protein is made constant and frictional forces are different for different molecules. Sodium dodecyl sulfate is an anionic detergent. It has sulfate at one end, which carries negative charge, while the other end is highly hydrophobic. When SDS comes in contact with proteins in presence of reducing agent like beta-mercaptoethanol or dithiotritol, it binds to hydrophobic regions of protein and provides net negative charge on protein molecule. The binding of SDS to per unit length of protein molecule is almost constant for large number of different proteins. For every two residues of amino acids, one molecule of SDS binds. This brings charge to mass ratio almost constant for most of the proteins. Since binding of sodium dodecyl sulfate to per unit mass of protein is constant and net charge on protein is negative. Protein sodium rhodocyl sulfate complex will move toward anode in an electric field. Also, charge to mass ratio of protein in SDS space is constant. Therefore, force exerted on protein molecule in an electric field is constant. In SDS space, mobility is determined by frictional force exerted by support on protein molecules. Sodium dodecyl sulfate protein complex moves through the pores of acrylamide gel and friction is experienced by complex while moving. The quantum of friction is different for different molecules. Relatively large size molecules will experience high friction and thus move slowly. On the other hand, small size molecules will experience low friction and will therefore move speedily. Depending on the size of the molecule, the value of friction will vary and ultimately it will be reflected in velocity. Also, it is to be noted that if size of protein SDS complex is larger than pores of gel, the particles will not move and particles will remain stagnated at entry point and these particles will not be separated. Similarly, if size of all particles is much smaller than the size of gel pore, all particles will move speedily and close to die front. These particles will also not be separated. The ideal situation demands that protein molecules in mixture must experience frictional force different in magnitude to allow their separation. In case either desired protein is not separated from rest of the protein or all proteins are not resolved in gel, gel of altered pore size is required to be used. Protein molecules after binding to SDS unfolds and SDS space is carried out in unfolded or referred as denatured state to have all molecules in unfolded state. Beta-mercaptoethanol or dithiotol reduces the disulfide bonds and therefore assists in complete unfolding of proteins. Oligomeric proteins after reduction with beta-mercaptoethanol or dithiotol in presence of sodium dodecyl sulfate will exist in monomeric form. Frictional force will be dependent on size of the molecule and it will determine velocity. SDS space is carried out in discontinuous buffer system. In the system, buffers used in tank and for preparation of gels are different. Electrode buffer is trisglycine of pH 8.3. Stacking gel buffer is of pH 6.8 tris HCl. Separating gel buffer is of pH 8.8 prepared from tris HCl. Further, separation requires migration of particles first through large pore gel, which is referred as stacking gel, and then in small pore gel, which is referred as separating gel or running gel. Stacking gel is layered over the separating gel. Upper end of the stacking gel and lower end of separating gel remain in contact with electrode buffer during electrophoresis. Lower molecular weight proteins move faster than high molecular weight proteins. Lower buffer tank is connected to anode while upper buffer chamber is connected to cathode. When electrophoresis is started, ions from stacking gel buffer, that is tris HCl, ions from tank buffer, that is tris glycine, and protein SDS complex start moving into stacking gel. Chloride ion from tris HCl leads, while glycinate ion from tris glycine buffer trails, and protein SDS complex moves between these two ions. This leads to concentrated protein in thin zone referred as stack. The pH of stacking gel is 6.8. At this pH, net negative charge on glycinate is low. Therefore, glycinate ion trails in stacking gel. The protein molecules continue to move in stacked gel until they reach the separating gel. When glycinate ion enters separating gel, which has a pH of 8.8, it acquires more negative charge, resulting in enhanced mobility. In separating gel, glycinate ion will cross protein SDS complex. The purpose of stacking gel is to concentrate proteins before their entry into separating gel. Acrylamide concentration in stacking gel is kept low, around 4%, while higher concentration of acrylamide from 7 to 15% is used in separating gel. In stacking gel, all proteins do not experience friction from pores of the gel. Acrylamide is prepared by polymerization of mixture of acrylamide and methylene-based acrylamide using free radical reaction. Free radicals are generated by decomposition of ammonium persulfate by tetramethyl ethylene diamine. Tetraethylene diamine exists in free radical form and it catalyzes conversion of persulfate to generate sulfate-free radical. Aqueous solution of ammonium persulfate can also generate sulfate-free radical, but this process is fastened by timide. Sulfate-free radicals snatches one electron from carbon-carbon double bond of acrylamide or bisacrylamide and later becomes free radical. Acrylamide or bisacrylamide free radicals will act on new molecules of acrylamide or bisacrylamide, and in this way long chains of polymers incorporating many molecules of acrylamide and some molecules of bisacrylamide are synthesized. Bisacrylamide forms crosslinks between linear chains of acrylamide, and therefore bisacrylamide is used in low amount in reaction mixture. Now I will be discussing how the experiments of polyacrylamide gel electrophoresis is carried out. This requires certain equipments which include vertical gel unit, power supply which can be operated either in constant bowl or constant current options, then connecting lids, combs, spacers, glass plates. It is to be remembered that power supply and connecting lids are used correctly. Red coded color refers to the anode, while the blue or black color refers to the cathode. One notched glass plate is placed on flat surface. One spacer of thickness 1 millimeter or 0.5 millimeter or 1.5 millimeter. Each is then placed along each of the two edges so that spacer aligns with notch. Subsequently, rectangular plates is placed over it. The sandwich is held firmly between thumb and fingers. Sideways of both spacers were sealed with appropriate tape to overcome any possible gel leak during gel plate preparation. Plate is firmly placed on casting stand to allow sealing of bottom with sponge in stand. There is always the possibility of leakage at the bottom of the plate. The plate in standing position is then quickly placed in casting unit and screws were finger tightened. This is taken care by placing molten agar up to a height of 5 millimeter in trough for sealing the lower end of the plate with agarose gel. As I said earlier that in separating proteins through SDS-PAGE, it requires the migration of protein molecules first through the stacking gel and then to the running gel. For performing this experiment, running gel is first prepared, for which we require solutions of acrylamide bisacrylamide 30%, 4 times running gel buffer, 10% SDS, distilled water, ammonium sulfate and tamide. These solutions are mixed in sequence and with the help of dispenser, running gel mix is delivered to sandwich to a level of about 3 cm below the top of rectangular plate. Air should not be trapped while filling sandwich with running gel solution, and once the gel is about to polymerize, a small volume of water is layered over the gel. This prevents exposure to oxygen and also keeps the gel surface flat. In contrast to running gel concentration, which is variable from 7% to 15%, the gel concentration in stacking gel is kept low, that is something around 4%. This gel is also similarly prepared. It requires mixing of acrylamide bisacrylamide, stacking gel buffer, sodium dodecyl sulfate solution, distilled water, ammonium persulfate and tamide, and is layered over the running gel after removal of water layer from the top surface of running gel, and then appropriate comb which has the same thickness as of the spacer is inserted into stacking gel to make wells for the sample application. Once the gel is polymerized, then comb is removed at that stage so that the wells becomes available for the sample loading. When gel is ready for the electrophoresis, before starting of electrophoresis, one has to prepare the sample, and sample preparation is one of the important steps. It requires mixing of equal volume of sample with 2x sample buffer, and the contents are boiled for 5 minutes. Usually 5 to 20 microliter of the sample containing something around 1 microgram to 10 microgram of protein is loaded on each well of the plate. Electrophotic conditions requires use of constant voltage. Usual practice is to apply 50 volts when the die is moving through the stacking gel. Then voltage is enhanced to 100 volt till the die reaches the lower end of the running gel. The thumb rule is that electrophoresis should be completed between 2.5 hours to 3 hours. After electrophoretic run, a stacking gel is removed and separating gel is processed for staining. Once the electrophoretic run is over, next step to visualize the protein, which essentially requires staining of proteins in gel. For this purpose, staining solution is prepared by dissolving commercial blue to a concentration of 0.25% and prepared in methanol acetic acid water in a ratio of 40:7:53, and gel is merged in staining solution and put on orbital shaker for 4 hours. Subsequently, gel is placed in destaining solution, which is methanol acetic acid water mixture in a ratio of 40:7:53, and this destaining is carried out for 30 minutes. Afterwards, the gel is placed in destaining solution 2, which is methanol acetic acid water mixture in a ratio of 7:5:88, and orbital shaker is used for this purpose also, and when the bands become visible, the staining and destaining process is completed. SDS-PAGE electrophoresis is used for checking the purity of the protein sample, and on electrophoresis, purified protein must get one band only. It is only in case if the protein exists in oligomeric form and monomer units are of different size, then band corresponding to subunits will be there. This technique is widely used for determination of molecular weight of the proteins. For that purpose, molecular weight markers must be loaded in one of the lane along with the sample in other lane. Mobility of the die, mobility of the markers, and mobility of the protein in sample is calculated by measuring the distance of travel from the upper end of the running gel. Mobility and the log of molecular weight are plotted. One will get a straight line, and by measuring the mobility of unknown sample, one can calculate the molecular weight of the unknown protein. Dear students, we have covered the principle of SDS polyacrylamide gel electrophoresis. The underlying principles in SDS space is the difference in their molecular weight. Sodium dodecyl sulfate is anionic detergent, and when it binds to protein, it denatures the protein. If we also use the beta-mercaptoethanol or dithioethyltol, then protein is reduced. So on binding of the SDS to protein in presence of mercaptoethanol or dithioethyltol, the protein exists in denatured form. The binding of SDS to protein is uniform, and protein acquires the negative charge, and this will move in electric field towards the anode. Since the protein is reduced, if oligomeric protein is there, Then it will be reduced to monomeric form, and the molecular weight of the protein will be of the monomer rather than the oligomer of the protein. So it should be clear that in SDS polyacrylamide gel electrophoresis, the molecular weight will be of monomer rather than the oligomer of the protein. Also, we will be now discussing that what are the possible errors, what are the possible their remedies for successful performance of the polyacrylamide gel electrophoresis, specifically the sodium dodecyl sulfate polyacrylamide gel electrophoresis. So this will be explained in coming presentation in the form of helpful hints. In any of the technique, problems are always faced on the bench. In SDS space, I have listed these in the form of helpful hints, which will help in solving any artifact or possible errors or for the best resolution of protein in acrylamide gel. First one is that gel concentration is decided by the range of molecular weight of protein to be separated. So that means if molecular weight range is from 24 to 205 kilodalton, one should use 7.5% gel. Similarly, if the molecular weight of protein is in the range of 14 kilodalton to 205 kilodalton, then 10% gel. Similarly, for 14 kilodalton to 45 kilodalton, 50% gel is required. So concept is very clear that low concentration gel is used for separating high molecular weight proteins, and high gel concentration is used for separating low molecular weight proteins. Spacers can absorb the heat and thus lowers the temperature of gel at edges. If the gel is hotter in the middle than at edges, the mobility of dye front at edges will be lower as compared to mobility in the middle. Thus, during electrophoresis, run the electrophoresis either using cooling device or use low current. Another helpful hint is if gel is not polymerized properly at edges, current can leak down the edges, resulting in more mobility at edges. The solution could be not to fill more buffer in the lower end buffer. Also, air bubbles at the bottom of the glass plate can block the current flow. This will result in abnormal dye front. So that means if dye front is not uniform, one should look back whether any air bubbles are trapped at the lower end of the gel. Then remove it before you perform the electrophoresis. While placing comb in stacking gel, care should be taken not to allow air trap. Air inhibits polymerization and samples wells will be distorted. Also, one has to see whether all stock solutions required for the preparation are usually refrigerated temperature stored, and these must be brought to room temperature. At low temperature, polymerization inhibited. Oxygen also inhibits polymerization of acrylamide, and these solutions should be degassed before use. We have seen that many of the proteins becomes insoluble when protein samples mixed with sample buffer are boiled at 100 degrees centigrade for 5 minutes. When protein is precipitated, it will not enter into running gel or separating gel, and these will not be separated. So solution for this problem is that we may incubate the sample with sample buffer at 70 degrees centigrade instead of 100 degrees centigrade. Dear students, let us summarize what we have discussed today. We have discussed one of the very important electrophoretic technique, that is the acrylamide gel electrophoresis. A bifurcation of this particular technique is very popularly referred as the sodium dodecyl sulfate polyacrylamide gel electrophoresis. This technique is very widely used for detecting the purity of the protein. It is also used for the determination of the molecular weight of the proteins. Of course, when we want to determine the molecular weight of the protein, we must run the molecular weight markers in one of the lane of the acrylamide gel and measure the mobility of each of the molecular weight markers. Also measure the mobility of the unknown protein, and these values of the mobility can be plotted against the log of molecular weight markers, and a standard curve can be prepared for determining the molecular weight of the unknown protein. This technique is also a prerequisite for the Western blot. So wherever the Western blot techniques are used, SDS-PAGE becomes essential part of that particular technique. This technique relies mainly on the differences in the molecular weight of the protein or molecular size, and because of this, the frictional force exerted on the molecule in electric field is different, and that leads to the separation of the molecules in electrophoresis. Also, we have discussed today that how the pH of the electrode buffer, how the pH of the running gel buffer, how the pH of the separating gel buffer leads to the concentrating the protein molecules in stacking gel in the form of a zone. And the purpose of the stacking is entirely this only, that you have to concentrate the protein before they enters to the separating gel. And the separating gel is used to separate the molecules based on their molecular size. But you see that if stacking cell is not there, stacking gel is not there, then protein will not concentrate in the form and the resolution will be of much lower quality. This technique is now very widely used in protein laboratory and also in all laboratories connected to the life science. We see a great future of this particular technique, where it is expected that the every students of the postgraduate in biochemistry or in other discipline must be well aware of this particular technique so that they can use in their research programs wherever they work in any field. Thank you.